This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 428, recorded on February 10th, 2017. This episode of TWiV is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with free shipping by going to blueapron.com/TWiV. Hey everybody, I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in a sunny New York City, Dickson Despommier.
Hello, Vincent.
Post-snowstorm.
Yes, indeed.
It's rather cold, 0 degrees Celsius.
Do you realize that this was only the 11th time in 11 years that New York City has had to close school because of snow?
About once a year.
That's very unusual.
We don't have that much snow anymore.
That's the deal, isn't it? That's what I'm trying to say.
Only about 6 inches or so.
That's all.
Not a big deal.
And they still close the schools. In fact, they closed them in advance.
And also Columbia was closed.
Yep. Bunch of wusses.
Got an email from a student. Are you having office hours today? I said, no, if Columbia's closed, I don't do office hours. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's partly cloudy here, 30 degrees, but I would call it mostly sunny. And that means -2 Celsius.
You didn't get snow, right?
No, we didn't.
But you already have some on the ground, correct?
Not anymore.
No, it's all gone.
Yeah. We had a slight dusting yesterday, but it was hardly anything worth talking about.
Also joining us from western Massachusetts, Alan Dove.
Good to be here.
Birthplace, the birthplace of the nation. Sorry.
Birthplace of snow. Oh, no. Yeah, we're famous for revolting here in Massachusetts. So yeah, we got about a foot of snow in 6 hours yesterday.
Neat.
Right.
And then it continued falling, and we got a total of, 15 to 18 inches. It's hard to tell.
You got a lot.
Yeah, the wind came howling through and blew it up into drifts. So at some places it was 2 feet, and some places it was like 8 inches. So it was a little hard to gauge. But we got quite a lot of snow.
So do you have a Mount Snowplow out in front of your house?
Oh yeah. Mount Snowplow rose up overnight. And
How did you plow your driveway with the Wovel?
No, I have an electric snowblower now that I've been using.
Has it cord or is it chargeable?
It's cord. And the great thing about I all my outdoor tools, when I can, I get electric versions. So I have an electric chainsaw, electric snowblower, and I have a generator, which I got after our 2011 week-long blackout, little portable generator.
Yep.
And the problem with having that is, of course, we don't get blackouts very often, which is nice. But then the generator just sits around, and the engine sitting around will rust.
Mm-hmm.
Yes.
So having electric outdoor tools means every time I've got to clear the driveway or cut up a fallen tree or something like that, I whip out the generator, and several times a year I'm running it, and that keeps it in good condition.
So when you shoveled or when you plowed your driveway, you pulled out the generator and
I pulled out the generator and the it's a bit of a palaver to get the snowblower and the generator out, but the snowblower is very light and the generator is pretty light, and you set everything up and start up the generator and then just blow away the snow from there.
A bit of a palaver.
Yes.
Now, in a prolonged and idle discussion, to talk unnecessarily at length. Oh, that's what we do here at TWiV, don't we?
That's right.
Speaking of talking,
we do it well too. I must.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Are you palavering?
I'm listening to a lot of palaver, that's for sure.
Yes, you certainly are.
And I don't have to. There's no snow here.
No.
It is 75 degrees, 24 Celsius. I would say partly cloudy or mostly cloudy. There's a little sunshine peeking through right now.
Yep.
So it's good. It's all good.
It is all good.
I am still waiting for the 27th emailer to win the Emerging Infections book. This is going on a long time because 27th, was that December 27th? Can't be, but it feels like it was. Anyway, keep trying. We're not close. We are close, but not close. Right. Next time, I'm going to make it like the 4th or 5th. I'm doing it for the international folks
there you go.
who are not in the same time zone, but
Right.
nobody says, it's going to be too late.
Exactly.
We have a meeting to tell you about NIDO 2017, NIDO. This is being arranged by Ying Fang and Susan Baker. It's the 14th International Nidovirus Symposium, 4th through the 9th of June in Kansas City.
And there is nothing cooler than nidovirus.
Stop it. I just raised my hand because I want someone to tell me what a nidovirus is.
Why not? Coronaviruses. Nido means nest in Latin.
I see.
Okay?
I see.
They have overlapping
Nido.
mRNAs. But there are human and animal viruses in there.
I see.
And if you look at the program, which is online, we'll put a link to it.
Right, I will.
The speakers are actually from many different viruses, not just nidoviruses. Right now, they have online registration open, and the early bird deadline is March 3rd. I guess you get a break there. We're going to be doing a TWiV from the meeting.
Nice.
So check it out. This is an open, and the organizers, by the way, wanted me to say this is an open international meeting, as the meeting organizers recognize that the global community benefits from collaboration and sharing of ideas. Take that. Take that.
I wonder why they felt it necessary to specify that.
I know exactly why. And so do you, and so does everyone else
so does
listening.
Everybody.
Yeah.
Oh, boy. Anyway, check out NIDO 2017. Nidoviruses, Nest, Dickson.
Okay.
Coronaviruses.
All right.
Corona and artery viruses. That's the other one I was trying to think of. These are animal overlapping viruses. No, not viruses that are overlapping. You know what I mean.
Yeah.
Follow up.
It's interesting. Viral zone
Yes, sir.
does not list it. This must be a new order.
It's an order for sure.
Yeah.
They don't list it?
Nidovirales.
Nidovirales.
And viral zone is not up to date on this, as far as
Really?
I can see.
Oh,
That's unfortunate.
they still have them listed as nifty viruses.
Here you go. Nidovirales, positive strand RNA viruses. Dude, I got it right here.
You do?
Yeah. Click on the plus RNA from the first page, and you get to order Nido.
I've been doing search virus or whatever. Okay.
Order Nidovirales. Yeah. Okay.
One of 7 genomic arrangements.
You
by
bet,
the way,
Dickson.
I might add.
What's the polarity of the genome of a Nidovirales,
You said
Dickson?
plus.
Hey, good.
Strand.
And you listened.
You just said it.
Yeah, but you listened.
But I don't have that short
Because
term.
usually you don't listen.
That's not true.
Follow up from Andy Ball. Many thanks to you and your colleagues on last week's TWiV for your thoughtful and generous presentation of my 1976 PNAS paper on VSV transcription. My co-author, Carol Norris White, said, how cool is that? You know, it doesn't get any cooler than to be on TWiV. Just for the record, the description of the paper's background that I sent you was indeed a TWiV exclusive, whipped off just for the occasion, 19 references and all.
Nice.
I would cherish a TWiV mug to remind me of my 15 minutes of fame. You bet. You got it. One TWiV mug in the mail. Now, I wanted to... I lost sleep this week because I fear we didn't explain one thing clearly in the discussion of that paper. I don't know if any of you ever worry about this. Probably not. But, after a TWiV is out, I'll go, we didn't explain that well. And I don't want someone somewhere saying, I don't get this. Why did I focus on one hit? Why do we care if the infectivity is, there's 37% of infectivity remaining, which means there's one hit. Well, we only want one UV hit per genome, because if you have more than one, you will not have a correct estimate of the target size, right? So you can imagine you would be putting in an excess of UV. You'd still be inactivating, but you don't want to put too much. You want one hit on average per genome, okay? Now I got that off my chest.
Yep. Because if you have multiple hits per genome, the point was to try and figure out what's connected to what.
Yep.
And if you hit 10 times in the genome, there's no telling what you hit.
That's right. Kathy,
I slept soundly throughout that entire week.
Good.
I thought you were going to say throughout that entire episode.
No, that too, of course.
He doesn't worry about it. That's fine. You don't have to. That's the way I'm wired. And I feel it's same thing lecturing to students. If I forget to say something the next time, I bring it up.
I'm that way when I do.
You got to do your job.
Absolutely.
Some of us.
What I worry about more is the stuff I forget to say and don't remember that I forgot to say it.
Yeah, that's the unknown unknowables, right?
Yep.
Unknown unknowns. Anthony writes, he just joined ASM. You're 100% responsible for my joining ASM. Indeed, I didn't know that there was an ASM before having the great good fortune to learn of Microbe TV podcasts. Not that I'm anyone in particular. I'm sure that I'm just one of the many who've joined ASM because of your tireless efforts. You are you, Anthony.
Yes.
You are a human, which is amazing. You have thoughts. You are you. You are someone in particular.
Listener, which is great.
ASM, of course, is the American Society for Microbiology. Thank you, Anthony. Anthony is very engaged. He writes to all the podcasts. Now, I received a correction email from someone who did not want to be identified nor have the email read, but it was stressed that I should make this correction. And this is having to do with a couple of TWiVs ago where we talked about cervid prions, chronic wasting disease. And the writer wrote that the reason chronic wasting disease is a big deal is that cattle are susceptible. They get it from deer in the pastures, and then it can enter the food chain. This is not correct. Chronic wasting disease is not known to cross from cervids to bovids under natural conditions. Any concern for the human food chain would relate to consumption of deer, not beef. And I stand corrected. What I referred to was if you inject cervid prions into the brains of cattle, they will get infected. So there's no... Right. And that's important because that demonstrates there's no species barrier because there are barriers among different species. But if you just feed cows deer prions, well, first they say, I don't want to eat deer, and then you talk them into it, and they don't get chronic
They say,
poison.
no, thank you, dear.
That's right.
So it's less likely that you're going to get transmission from deer to cattle. But certainly hunters have to be careful because they could get it from deer. But so far there has not been a single case of deer human.
Yeah, it's exceedingly unlikely that this transmits to humans because we've known about it since the 60s, and there hasn't been any documented case of any, hunter transmission of it.
Yeah.
And I think also since the deer with this tend to go downhill really, really quickly, they're probably not targets for hunters.
Yep.
I don't know.
But I still think it's a cool disease. Although
it's a fascinating
the deer
disease.
may not think so.
No.
Or the elk.
You know why I don't like deer lately, Dickson? And
over your
and everyone here
eat all your landscaping.
Not only in my yard, I wouldn't care so much about that. Every time they walk by the window, the dogs start barking, and I can't record podcasts at home because you want to have this dog barking in the background. Oof.
I don't know. It would make it kind of homey. It's good.
Yeah.
We got it. We have, the equivalent of humans barking in the background. I don't know why we shouldn't have dogs.
Okay. We're palavering in the background, aren't we?
Yes.
All right. We have a snippet which
Labrador Retriever.
I'd like to tell you about, which is pretty cool, published in MBio. And this is a snippet. We're going to do this in 15 minutes.
Or less.
Yeah, or less. Novel superspreader bacteriophages promote horizontal gene transfer by transformation. This is by Eric Keene, Valerie Bliskovsky, Malagon, Baker, Prince Klaus, and the senior author is Sankar Adya, who I have known since I was an undergraduate as a phage person. Not have known him, I've known of his work. Oh my gosh, he's a famous person in phage. So here's the shtick with this paper. There are 10 to the 31 phages, bacteriophages on the planet. Oh, that's a lot. If you put them end to end, they would stretch 200 million light years.
That's a Dorothy Parker's worth of phages.
Well, I don't know who Dorothy Parker is. Who's Dorothy Parker?
She was a member of the round table at the Algonquin Hotel, and I'm sure that Alan can regale us in some of her quotes.
Anyway.
I'll pass for the moment.
All these phages carry out between 10 to the 23 and 10 to the 25 infections of bacterial cells every second.
A mole's worth.
Yeah,
A mole's
10 to
worth.
the 23,
Yes.
a mole's worth.
Avogadro
There you go. There's our title, a mole's worth of infections. And, of course, when a bacteria lyses out, spews DNA. And so this paper is saying, can that DNA, is it intact? Can it go into another bacterium? Now, we know that phages bring DNA from bacterium to bacterium by a process, Dickson, called
transformation.
No, transduction. Transformation is naked DNA getting which that happens also. Okay. So they want to know here, can phages do this? So they have a beautiful, simple experiment. And you know how I can tell if an experiment is simple? I can read the paper in 5 minutes and understand it. All they do is they take a E. coli host with a plasmid in it that encodes ampicillin resistance. And I'm already on this because I use these for years, these plasmids, right? They infect them with phage, and then they extract DNA from the lysate, the bacteria Lyse. They extract DNA, they make competent cells, and then they put the DNA and they ask, do we get any antibiotic resistance? Really simple. You plate them on ampicillin-containing plates, okay? So what do they use for phages? T4 and T7, and I thought Rich Condit would like this.
Yeah, I'm just drooling over T7.
And then they had 20 phages that they had isolated from soil and water and feces. And here's the most important part of the paper: Miami and Washington, D.C. No, I'm just kidding. But they're lytic phages. And it turns out if you do this assay with all these, two of them stick out. They have thousands of times more efficient ability to release intact plasmid that can then transform antibiotic resistance into E. coli. It's amazing. Lovely table, lovely graph, which you can see all these isolates. And T4 and T7 don't do very well. They chop up the genomes and the plasmids, presumably. Right. And these two phages, so they call them super spreaders, SUSP1 and SUSP2. It's very cool. And these super spreaders, they're not specific for this one particular plasmid that they use. They try some other plasmids and they work as well. And it's not transduction. They check that. The transduction is when a phage picks up a piece of DNA from the host genome into its own and moves it to another cell. It's not that. It happens at low multiplicity of infection, so it's not simply the phage is lysing the cells initially. And what they're doing is the phages, they look at the DNA that's being released, and these super spreaders lead to the release of intact plasmid DNA. The other phages that don't do this, they chop it up, which is really interesting.
Right.
Now, these two phages, they're novel, so they sequence their genome and they find that they're missing known hydrolytic endonucleases that are involved in chopping up DNA. Okay?
One question that people might ask is, why do the phage chop up DNA?
Rich, I was just going to ask you that.
If you pre-label E. coli DNA with, some sort of radio label, so you pre-label the host DNA,
right,
and then infect it with bacteriophage T7, the progeny DNA in the bacteriophage T7 is labeled. The phage presumably degrades the host DNA. This is anthropomorphic, but degrades the host DNA for the purpose of using that DNA in its own progeny genomes.
It scraps it for parts.
That's right.
Scraps it for parts.
Melts it down and repours it.
But then if it doesn't have functional endonucleases, it results in the release of all this intact DNA, which then gets into adjacent bacteria that have not been infected yet.
Yep. Now, Rich Condit, they have a mutant of T4 that also promotes plasmid transformation, and this is missing an endonuclease as well. Right. Does the absence of these endonucleases exert a fitness cost on the phage?
I do not know the answer to that. I was wondering the same thing myself. It's probably out there in the phage. I'm sure it's out there in the phage literature because the mutants are so well characterized, but I don't know the answer.
Let's get back to that discussion after we talk about the interspecies transfer, because I thought this was a really cool aspect of it.
So next they said, let's get away from E. coli. They went and they got dirt from Bethesda and Cheyenne. Bethesda, Maryland, and Cheyenne, Wyoming. Hey, near Saul Silverstein's house, right? Cheyenne is not near Jackson.
Nah, it's right on the border.
Just they take soil, they just wash it through with medium and get the bacteria out, and they grow them up, and they simply add lysates that they've made in these other experiments from the super spreaders. And in fact, they can introduce resistance into these soil bacteria by these lysates. So it's not as species specific. And in fact, they identify one of the bacteria as a bacillus species in these experiments. So it can go, is a phage that normally infects E. coli, and the lysate can transform DNA into bacillus.
Right. So I found this to be the most convincing aspect of the argument that the transfer is not by transduction. It's not by the phage carrying in the DNA inside the phage particle. Because the experiment where they actually first described that in the paper, when I read it, I wrote beside it, I find this not so convincing, because it was a little bit of, to me, it was like negative data. I wanted something more positive. And so this fact that the transfer occurs into bacillus, which the phage can't themselves infect, I found much more convincing. What they did is they spun down the supernatants to remove the free phage, and that reduced the phage titers by roughly 1,000-fold.
Okay.
But the supernatant was still transformed as much. And I just wasn't so convinced by that.
Yeah, this is a good one, of course, because the phage can't infect these bacillus species.
Right. So the resistance is being transferred by the bacillus salvaging bits of the E. coli that got lysed.
Right.
Which it can only do because these particular strains of phage lack the nuclease.
Yep. So that's the paper. It's a perfect snippet. Interesting implications, because there's moles worth of infections per second going on. A lot of DNA, as Eric Delwart said, DNA is everywhere. And
what I love this line in the discussion: since the virosphere comprises an estimated 10 to the 31st phages, of which we surveyed 20, it's exceedingly implausible that these two are unique.
It's very good. I highlighted that too.
Yeah,
it's very good.
What I think is cool here, this is speculative, of course, but you've got these strains of the phage that have this endonuclease, which, as Rich explained, seems to be there to chop up the host DNA, which probably helps in the replication of the virus. And then you have mutants who don't have the endonuclease, and that would seem like a disadvantage, but there's a potential upside to it in the natural environment, which is that by leaving the DNA intact, when that cell lyses, it's going to spread whatever useful genes were in there, and bacteria are very good at picking up useful genes, to the whole neighborhood. And so subsequent hosts that the phage might be able to get into are going to benefit from this, spreading of the useful genes. It's almost like a difference between a short-term strategy and a long-term strategy for the phage. Right. So the short term would be get in, replicate fast, cut up everything in sight, and build your genome out of it and get out. And who the heck cares what happens next? Whereas the long term would be get in, replicate, but don't cut up the host DNA, because then when you finally lyse the cell, it's going to potentially give you a broader, more resistant community that you can then go on and infect.
Yeah. So you're saying evolution is for the future?
No, what I'm saying is that evolution could select for two different strategies. They wouldn't be strategies in the sense of the phage thinking through it, but you could select for a phage that replicates very fast in a destructive fashion that may not be able to last in a population that's put under something like an antibiotic stress. And you could also select for a population that replicates more slowly, that would have a disadvantage if there's no antibiotic, but it would have an advantage if there is an antibiotic, because it has helped to propagate the antibiotic resistance.
But it hasn't helped that particular phage, though. It helps the next generation, right?
It would help that next generation of phage, which is a big part of evolutionary selection, because then that phage could then go on and overgrow the whole population.
Maybe. Could be. I understand what you're saying.
Yeah,
I see that.
Anyway, I thought I agree this is a very cool paper.
So here I have a 1970 paper in the Proceedings of the National Academy of Sciences by Melvin Center, F. William Studier, and Charles Richardson, entitled "The Structural Gene for a T7 Endonuclease Essential for Phage DNA Synthesis." So if you knock out the gene in T7 doesn't make much DNA. So yes, there is a fitness cost. As a matter of fact, they got AMR mutants in this. So that means that's essentially an essential gene. Yeah. And I think there is another nuclease as well, which is the gene 6 product. I'm just kind of skimming this, but I presume it has a similar sort of phenotype.
Now,
So,
of course,
yes, there's a fitness cost.
These sus phages, their genome's not been annotated, so we don't know if there aren't other proteins that might serve the same functions, whereas these endonucleases are dispensable.
Mm-hmm.
They don't chop up the genome, but if there is a nuclease required for DNA replication, it may be in some other protein that we don't know about. But I think this image of DNA, plasmid DNA pouring out of cells every second to huge numbers is really compelling.
Yeah.
Tell out there, bacteria are picking it up, and eukaryotes can pick it up too. Why not? Interesting. And just to continue an arc, eukaryotes didn't invent anything.
Why would we?
It all came from bacterial DNA. Why would we?
Why invent it when you could just steal it?
Exactly.
I had occasion to talk about that in class this week because, of course, E. coli, and I can't remember if it's other bacteria also, but they polyadenylate their RNA. So,
When this phage chops up the host DNA, does it chop it up into single nucleotides or little snippets, little pieces?
What a good question. So, as Rich said before, it can do nucleotides, but they didn't experiment with this T7 mutant, and they say it doesn't go to nucleotides. It went to 100 MERS roughly.
So a lot of it would hybridize with the host DNA?
Why would it hybridize?
Because it's the same.
It's a plasmid. It's a bacterial plasmid.
Yeah, but it's got the same sequence though, right?
No, it doesn't have the same.
I mean, portions of it have the same sequence.
No, this is phage. I'm sorry, plasmid DNA being released. What's going to hybridize with? Sorry, I don't get.
No. Okay.
No, these are these sus phage, these super spreaders are double-stranded DNA phages, and they chop up. Normally, phages chop up the host, as Rich Condit said, so they can use the triphosphates.
Right.
But in this paper, there is one experiment where they say it doesn't go to triphosphates for some reason, but most of the time it does.
Okay.
Anyway, why did you ask, by the way?
I don't know, just out of curiosity.
How many different kinds of
7.
All right. So there's an $89,000 PCR machine in this paper?
Oof.
They do a thing called
drop digital PCR.
Yeah, drop digital PCR, which I was unfamiliar with. So I did some research on this, and it's fascinating. They basically, it's to me, a new way of quantitating the amount of a small amount of a specific DNA, like mutant or type or plasmid, in a complex mixture. And it involves a machine. You take your DNA, and what they do here is they make an oil emulsion. The machine does, and uses microfluidics so you get little oil droplets that contain a few fragments of DNA out of your DNA sample. And the machine then robotically disperses these into a 96-well plate. But each well is going to contain probably hundreds, if not thousands, of these droplets. And you do PCR on those, and it's contained in the droplets. And the way the PCR reaction is set up, if it contains the DNA of interest, that droplet gives fluorescence, and it is positive. If it does not contain the fragment of interest, it is negative. Okay. There's then another machine that this goes through that sorts out the individual droplets from each of the initial wells and scores them as either positive or negative. So in the end, you can have like 20,000 droplets that you've done PCR on in a 96-well plate using this robot, and it counts the distribution of positives or the number of positives in that sample. And then use the Poisson distribution to tell you, the probability that any given sample had 1 or 2 or 3 DNAs in it. And you get that from that, you get the ultimate concentration of DNA. So you can't do long sequences. You can look for sequences up to 200 nucleotides in length, but you can quantify very accurately rare species in a complex mixture of DNAs doing this. And this whole digital PCR thing, or drop digital PCR, is well, according to the advertisers at any rate, becoming the rage. So I had the link I have the link I have there is to a YouTube video that's very nice and describes it. And I'm always interested in how much, right? So the machine from Biorad costs $89,000, and it's $3 a reaction.
How can it become a rage at that price? It's ridiculous. They would like it to become a rage. Maybe the price will drop.
Well, maybe they were using the word rage differently than we are.
Well, the economics of a business like this are if you sell 8 of these, it's a rage.
The advantage of this is that it allows you to quantitate your product on an absolute scale rather than a relative scale.
Right.
Yeah, but it's too expensive. You have one.
Prices will probably come down. No, we don't. At least not that I know of here.
This would be a shared facility type.
Yeah. This is a core. Yeah. This is a core facility purchase.
Although I bet I know people who would have one of their own, even 2, absolutely, and they have too much money.
One for each strand of their DNA.
This episode is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everyone, while supporting a more sustainable food system, setting the highest standards for ingredients, and building a community of home chefs. And I have the Mr. Sustainability here in front of me. Dickson Despommier, the father of the vertical farm. You don't have to say anything.
I didn't.
Blue Apron delivers seasonal recipes along with fresh, high-quality ingredients so you can make delicious home-cooked meals. I know. I've made some. They're really good. Each meal comes with this card with step-by-step, easy-to-follow recipes and pre-proportioned ingredients. They give you everything except salt, pepper, and oil. You can do it in 40 minutes or less if you spend a lot eating out or at high-end grocery chains. This will cost you under $10 a person for healthy home-cooked meals. Really good. Now, they get their stuff. They get their foods from their community of over 150 local farms, fisheries, and ranchers, including seafood, beef, chicken, and pork, which the latter come from responsibly raised animals, produce from farms that practice regenerative farming, and they give you exactly what you need for a recipe, so there's no wastage. Now, I like to eat. I don't like to cook so much. And people always say to me, how can you not like to cook? Because you're a scientist. You're always mixing things. And I say, I never put a pinch of anything in my tube at work. It's never a pinch or this or fold in. It's very specific. So I don't get this analogy between cooking and science. However, I like Blue Apron because it's all laid out there and there's no pinches. Everything is measured out. You just put it in. You can customize your weekly recipes. They have, of course, vegetarian. Get your delivery options whenever you need it. They go to 99% of the continental U.S., and they don't repeat their recipes within the year, so you don't get bored. Here's some recent recipes. Cashew chicken stir-fry with tango mandarins and jasmine rice. Crispy barramundi with quinoa and roasted carrot salad. We talked about quinoa once on Urban Ag, that old podcast we used to do. Udon noodle soup with miso and soft-boiled eggs. Wow. Check out this week's menu. Get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiV. That's blueapron.com/TWiV. You will love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. Don't wait. Blue Apron, a better way to cook. I vouch for it. It's good. I've tried it several times, and I shall try it again. We're going to do a paper now. It was published in February in the Journal of Clinical Investigation. Regional astrocyte interferon signaling restricts pathogenesis during neurotropic viral infection. And this is out of Washington University, Cal State Pomona, University of Washington in Seattle, University of North Carolina. And the first author is Brian Daniels, and the last author is Robin Klein. And also on this paper are Helen Lazier, who's been on TWiV a few times, Michael Gale, and Michael Diamond, all of whom have been on TWiV. And so here's the history of this. A couple of weeks ago, we were talking about astrocytes, and Kathy was talking about their function in the immune response. And then this paper came out, so she had put in a follow-up to last week's about it. And I saw the paper too, and I said, we have to do this paper. So we're doing it. And this is about the role of astrocytes in pathogenesis of a neurotropic viral infection. And the virus is West Nile virus, Dickson.
So it is.
You can wake up. Do you know what polarity its RNA genome is?
Think positively.
Thanks for the hint, Kathy.
What family is?
It's a flavivirus.
What other flaviviruses do you know of?
Let's start with yellow fever.
What else?
Japanese bee encephalitis.
What else?
Come on,
Lacrosse virus.
What's the big one, Dickson?
West Nile virus.
The big one.
Dengue fever.
You're always.
That's not the big one. that's the one at the end of the alphabet.
Zika. It goes from. Does it go from A to Zika? No, not the family. There's no A.
No, it's not. Adeno's
No, that's not a flavivirus.
I know it's not. I know. That I know.
But if you wanted to, you could go from adenovirus to Zika virus. That's what Tony Fauci does when he gives a talk. So what we want to do here is look at the role of astrocytes, not Zika. This is back to West Nile. Neurotropic virus. How many people get neurotropic infections?
Many.
What percent of total infections? 10, 20, 30?
I wouldn't know the answer to that.
Okay, even though you wrote a book on it.
Well, you mean how many people that get West Nile get a neurotropic West Nile?
Yes.
It depends on the strain of the virus.
Okay.
And, pardon me. The United States, there appears to be a strain that's more neurotropic than the strain that it was derived from in Israel.
Okay.
So
there's the model they use here is infection of mice. They put virus in the foot pad, it's subcutaneous, and then gets into the CNS. And then they also do some intracerebral inoculation. But they have a nice mouse model for infection. And the point of this paper is to figure out the role of astrocytes, and they want to know the role of the interferon response in astrocytes. And because previous work has shown that astrocytes have some role in pathogenesis via the interferon response, and one of them is via regulation of the blood-brain barrier, BBB. It's not bed and breakfast. It's blood-brain barrier,
and it's not the Better Business Bureau.
No.
Which protects the CNS. And this blood-brain barrier, it's a real thing, Dickson. It's made up of specialized cells. They're called brain microvascular endothelial cells.
Yeah.
The little capillaries that are in the brain. They're joined by tight junctions. They're covered with pericytes and the end feet of astrocytes, and that makes up the blood-brain junction. And it's been shown that if interferon signaling contributes to the integrity of this junction, that's what this paper is all about. This
is a critically important junction in the body because it keeps it's responsible for keeping all the bad stuff out of your brain.
Yep.
So the reason we don't have lots and lots of neurotropic infections is because of the blood-brain barrier, because most viruses simply cannot get through it.
Right.
Now
it blocks molecules that are as small as 400 Daltons.
Mm-hmm.
Yes,
molecular weight of 400.
It's a very, very tight barrier, but it still allows nutrients and oxygen through. So it's a selectively permeable barrier that is not nearly as well understood as we'd like.
It also has transporters that can allow transport of things.
Yes.
So when you get an infection, though, you get inflammation, and this can compromise the blood-brain barrier. Not only the brain, but in other tissues as well. But as far as the brain goes, you get the production of inflammatory cytokines, things like TNF-alpha, interleukins of various numbers. They disrupt the integrity of the blood-brain barrier, and this is being studied. Okay, but part of it involves interferons. The cytokines allow leukocytes to get into the CNS because, if you have an infection there, the leukocytes want to get in and clear the pathogens, but by loosening the blood-brain barrier, you're letting virus in at the same time. And this also happens in the respiratory tract and the gut tract. You have a basement membrane underneath the layer of epithelial cells. It's a nice barrier to virus, but as soon as you have inflammation from a virus infection, these leukocytes start migrating out and the viruses go in. Similar idea.
And it's, evolutionarily, you need to make that barrier permeable to get the leukocytes out, but that opens you up to invasion by the viruses, so there's probably no good solution to that.
Right. Now, so these cytokines, these inflammatory cytokines loosen up the barrier, but type 1 interferons are believed to stabilize the barrier. They stabilize the integrity of the BBB, they enhance tight junction formation, and it's sort of the antagonism of the inflammatory cytokines. So type 1 interferons remember this. It's important. Otherwise, we're going to get confused. They maintain the barrier, and the inflammatory cytokines break it down. Now, what's this have to do with astrocytes? So astrocytes can sense foreign substances by pattern recognition receptors, and they can then produce cytokines and type 1 interferons, and these, of course, can prevent or prevent infection, but they can also exacerbate infections as well. Now, the interesting thing here is that they're different throughout your brain. They're different in terms of gene expression, morphology, how they develop, how they signal, and they respond differently depending on what part of your brain they're in to pathogens. And that's very cool, and that's something we're going to get into in this paper. So the question here is, how does interferon signaling? Now, remember, interferons are produced in infected cells. They then bind to cell surface receptor, interferon receptors. They stimulate a transduction pathway that then leads to the production of interferon-stimulated genes, which have antiviral activity. And what they're asking here is, what's the role of the interferon response in astrocytes in mice infected with West Nile virus? And when you infect mice with West Nile, the neuron's the target of the virus who replicates then in them. You do get activation of astrocytes in these infected mice, and they would like to know, what's the role of interferon signaling in astrocytes? What does it do to the blood-brain barrier, etc.? Now, the cool technique that they use is they can knock out the interferon receptor specifically in astrocytes, and in specific astrocytes as well. So this subset of astrocytes are positive for glial fibrillary acidic protein, GFAP. Okay? So what they do is they take the interferon receptor gene in these mice, they flank it with LOX sites. They flox it. And those can be cut out by a recombinase called CRE, which they make under the control of the GFAP promoter. So it cuts out the gene. You make transgenic mice, when they're born, they lack the IFNAR gene only in these special astrocytes. Nowhere else.
Because these astrocytes would activate the GFAP gene, which also turns on the CRE recombinase, which then cuts out.
Yeah. Now, the cool thing is, now IFNAR, these are IFNAR mice, you can make mice with the gene knocked out everywhere, in every tissue, and they're fine. Right. I mean, they live, and you can infect them, and we have done it, and many other people have done it as well. But here they just want to know in astrocytes, and it's a specific subset of, it's not every astrocyte as well, but it does bring down IFNAR in these cell populations. So you delete, if you take IFNAR away globally, the mice get very sick and die very quickly. But if you take them away from astrocytes, all right, and they confirm that IFNAR is very low in the cerebral cortex and the cerebellum. We're going to look at those two areas mostly in this. Are you okay so far, Dickson?
Absolutely.
Ask a question. Raise your hand.
No, I'm absorbing the material first.
So they confirm that the deletion of IFNAR is in these astrocytes as they
He's allowing the information through the DDD barrier.
I am. Thank you.
DDD barrier.
Thank you.
So, microglia, neurons, etc., they still have IFNAR. All right. So the subcutaneous 10 to the 2 PFU of West Nile virus. Dickson, the New York strain. Isn't that great?
Yes. The 1999 strain.
2000.
Okay.
Must have got it from the following year. Right. Now, these mice with IFNAR knocked out specifically in astrocytes have accelerated and enhanced mortality and greater clinical signs of disease compared with controls. No differences elsewhere in viral burden. So it's just the CNS phenotype. But in the CNS, the virus levels are higher in these, what do we call them? IFNAR null astrocyte mice, right? Just lacking IFNAR in the astrocytes. And they also find virus earlier in cerebellar tissues compared with controls. By day 6, viral burden is higher in the cerebellum, and titers in the cortex and brainstem were elevated, but not statistically significant. So what's happening here, cerebellum titers are going up higher and faster after knocking out this IFNAR in astrocytes.
So I'd just like to insert a little bit here, because not really having been that familiar with brain anatomy until we started working on mouse adenovirus and found it in the brain, I think that other people might not always have all those parts of the brain straight. And also in this,
I don't even have the parts.
in the discussion, instead of talking about the cerebellum versus the cerebral cortex, they start talking about hindbrain versus forebrain. And so the forebrain in this case, and most of the experiments where they don't see as much of an effect, is the cerebral cortex, right. And the hindbrain where they see this effect of the IFNAR mutation in the astrocytes is in the cerebellum. So those words all begin with C, but just try and keep them straight as you keep listening.
So
and you think with your cerebral cortex and you walk with your cerebellum.
Very good. I was just about to ask that. Where the emotion part in the cerebrum?
I think
that would be
amygdala.
Amygdala.
Well, right. I mean,
fear and anxiety map to the amygdala.
Yeah.
How about fear? How about fear and loathing? Where does that happen?
That starts at an early age.
Yeah.
The
amygdala and adjacent areas often described as driving the 4 Fs. And that would be
There's a family
fight.
show. There's a family show.
Yeah, fight, flee,
right.
Fright, and mate. Yeah.
Right.
And mate. That's right. Thank you, Alan.
So let me get this straight. Hindbrain is equivalent to cerebellum.
Yes.
Yeah, that's right.
Yes.
Forebrain is equivalent to cerebrum.
Yes.
Right.
Okay.
Right.
At least within the confines of how this discussion
within this discussion, right? Forebrain refers to the whole front end, which includes the cerebral cortex. Hindbrain refers to the whole back end, which includes the cerebellum.
Kathy, are these your diagrams here?
The one is from a Nature Reviews, the one that's just the map of the parts of the brain, and then the other is from our review on the blood-brain barrier. Yeah.
Okay. It's really, these are really nice. They're very helpful.
Yes.
So it's interesting. It's interesting to me that the astrocyte, I don't know what you would call the projections off of the astrocyte, but they as at least according to your diagram here, basically surround the whole blood vessel.
Right. Those are the end feet. I think Vincent might have mentioned those.
Okay.
But
it's not just touching it. Do they actually coat the blood vessel?
As far as I understand it, yes.
Oh.
And it's those astrocyte end feet, plus probably the pericytes, that really make the tight junctions of the blood-brain barrier different from tight junctions in those other places that Vincent was mentioning. Yeah. And different, so there are also epithelial cell tight junctions that might be lung and gut and so forth. But even endothelial tight junctions elsewhere in the body don't have the astrocytes and pericytes.
So it's interesting to me, I mean, this maybe is a little, I don't know. It's interesting to me that in order to set off an astrocyte, something has to get through. It's on the outside of this whole show. Okay, so in order to tickle the the interferon receptor on the astrocyte, something actually gets through, and the astrocyte says, "Whoa, that's not right," and triggers this whole response. Okay, so it's really a guardian of this whole thing.
Yep.
Right.
Yeah, some viruses can do that.
Right. And then too, astrocytes have a role for neurons. And so you could imagine that might be different for a virus that gets to the brain through the neurons.
Okay.
Right.
So far what we have, we've taken out IFNAR and astrocytes. We see a phenotype which is more pronounced in the cerebellum after we put West Nile in. Earlier appearance within the cerebellum. So they say, all right, this is having anything to do with the blood-brain barrier. So they look experimentally to see if the permeability is changing in infection of the 2 different mice, the IFNAR knockouts, and they use a dye, sodium fluorescein, which they put subcutaneously. And if they let it get in, and then they perfuse the mice to take it out of all the vessels, and then they take the brain and they grind it up and look for fluorescence as a way of seeing if it's gotten in the tissues or not. And what they find is that they see an enhancement of permeability on day 4, then it goes down, and then it goes back up again on day 8. And they see this in both the cerebral and the cerebellar tissues, but there's different kinetics and it's more it happens sooner in the cerebellum compared to the cerebral cortex. So this is suggesting that the BBB its permeability is being altered, and this is again in the IFNAR mice compared to the wild type mice. There's a difference.
It happens in both, but it's more pronounced in IFNAR knockouts.
Exactly. All right. So this is suggesting that interferon signaling is involved in regulating the BBB, and so that interferon is a response to virus infection, so that could be involved. So then they say, does interferon signaling have antiviral functions from astrocytes? Bless you, Dickson. Oh, you didn't sneeze. Excuse me. Sorry. So what they do here is they put virus directly in the brain intracranially. So they're going to bypass virus getting through the BBB, right? Put it right in the brain. And what they find is viral titers don't differ between the two genotypes of mice in the cortical or cerebellar tissues when you put the virus directly into the brain.
I'm surprised, Vincent, that you haven't said anything yet about the fact that they're actually doing plaque assays.
Yeah, they are. PFU, everything is in PFU. It's wonderful. Yeah, I'm very happy
to see that. The difference they see in the knockout mouse or the Cre-induced mouse is due to the difference in the blood, appears to be due to the difference in the blood-brain barrier.
Right, because if you bypass it and inject it intracranially,
you don't see any difference.
Right.
In viral loads, right.
In viral loads.
Okay, so the summary here is that the higher titers in the cerebellum is due in subcutaneous infected mice. When you don't have IFNAR, is due to defects in the blood-brain barrier restriction of neuroinvasion. So you take away IFNAR, the BBB is opened up more, and it's letting virus in. Does this enhanced permeability of the BBB have anything to do with cytokines? They're called vasoactive cytokines, which would open up the BBB.
And this would be part of the inflammatory response opening up the BBB.
Exactly. Like type 1 interferons, TNF-alpha, IL-1B, etc. So they measure these at 6 days after subcutaneous inoculation. The cerebellar tissues of the knockout mice have higher levels of interferon beta and alpha compared with the controls. They have higher levels of IL-1B. Did I say that already?
No.
You said no.
I said interferon, IL-1B compared to the controls. And in the cortex, this IL-1B was not affected. There's no difference between the two animal lines in the cortex itself. By day 8, and this is all measured by mRNA, by the way. I guess it's RT-PCR. Mm-hmm. By day 8, mRNAs of these cytokines are higher in tissues from both the cortex and the cerebellum of the mutant mice, which they say probably is because you have higher viral titers there, and now everywhere these things are going up. So basically, the enhanced permeability of the BBB, which is accompanied by neuroinvasion, is at least in part due to changes in the expression of these barrier-stabilizing molecules. So the barrier-stabilizing are interferons, the barrier-disrupting are the cytokines. Are we okay so far?
Yep.
Good.
I think so.
Mm-hmm.
So they're tracing this to the cytokines. Yes, Dickson?
Where in this diagram is the choroid plexus?
Which diagram? In the show notes?
Yeah. No. This brain diagram here. I'm just trying to imagine the anterior and posterior choroid plexus. Isn't that where the brain barrier is?
That's a different barrier. That's the
CSF brain barrier. So that's where many viruses can get in because there is a pathway. That's the choroid plexus, if I remember, is where the CSF is produced.
Yes.
And so you have to be able to get it in there, so viruses can get in there.
Okay.
Right.
But I couldn't tell you where on that picture it is.
Yeah, I was having trouble. I also wondered whether it was the same permeability in mice that it was in people.
Oh, I see the amygdala down there.
Yes.
This is where fear and anxiety is, huh?
Exactly.
Can we take it out?
It's a lizard brain function. In some people, it has been removed.
All right, so they noticed that some inflammatory chemokines, two of them, CCL2 and CXCL10, were higher in cerebellar tissues in these knockout mice. So they wanted to know, is there more? These would be chemoattractants. They would attract cells. They want to know if there's any differences in infiltration of immune cells into the CNS. Now, if you look at the total number of infiltrating immune cells in the cortex, it's no different in the 2 lines of mice.
Yeah.
But there were greater numbers of CD8 positive, interferon positive lymphocytes, and also a certain type of macrophages. And I will not read them, all the markers for you, because you'll just get confused in the cerebellum. And in the cortex, no differences in infiltration between genotypes. Basically, you're seeing infiltration into the cerebellum. Cerebellum.
The forebrain or the hindbrain?
The hindbrain, the cerebellum, that little thing in the back.
The hindbrain. Right. The motor function. Yeah.
Type 1 signaling in cerebellar astrocytes is suppressing this infiltration of cells. Okay? Is this a pathologic or a protective response? How do they address that? They look at survival following intracranial inoculation. All the mice die when you do that, so there's no difference in overall survival, but the mutant mice lacking the IFNAR receptor in the astrocytes have accelerated mortality. So they all die in both groups after IC inoculation, but the ones lacking the IFNAR and astrocytes have accelerated death. And this is accompanied by CD3 positive lymphocytes. The CD3 is a general marker for lymphocytes, and also cells that have markers of microglial cells, macrophages of the brain, in the cerebellum. There's also apoptosis associated with these cells.
Now, the intracranial inoculations, they did them into the cerebrum and the cerebellum separately.
Right.
Mm-hmm.
And found that the cerebellum injected ones died faster.
Quicker. That's right.
In the mutants. The cerebrally inoculated ones died at the same rate as the wild type.
Yes. And that's why you have to say intracranial inoculation.
Yes.
Not intracerebral, which is what we used to say.
Just intrathecal, we used to say.
It's intracerebral versus intracerebellar.
Cerebellar.
Yeah.
And the intracerebellar is the one that kills the mutant mice faster.
Right.
Which indicates
go ahead.
Which indicates that the cerebellum is particularly susceptible when you've lost the BBB.
Also, that it's a protective response, because in this cerebrum, they're not dying any quicker with the absence of the IFNAR in the astrocytes.
Right.
And you've taken away that protection from the cerebellum, and that's why when you put the virus there, they die quicker.
So up until now,
yes,
we've seen increased virus in both the cortex or cerebrum and the cerebellum. And we've seen, well, I guess there were differences
Kinetics
in cytokine, and there were differences in cytokine production. Okay, so I guess this is consistent, that these effects are at least more pronounced in cerebellar tissue, all these effects, than in the cerebrum. Okay.
Yep. All right. Now, another way that type 1 interferon, a way that interferon modulates the influx of cells into the CNS is it modulates a cell surface protein called VCAM1, vascular cellular adhesion molecule. Okay. So they want to know if the enhanced inflammation they see in these knockout mice is associated with increased production of VCAM. So they measure mRNA of VCAM. So VCAM upregulation would facilitate immune cell trafficking into the CNS.
Right. This is like an integrin, right?
Yes.
That grabs, that is on, I guess it's on the astrocytes. Mm-hmm. Okay, but it grabs onto immune cells, or can.
Right.
Right.
Correct. So more of it, you get more infiltration. Right.
Right.
So they see greater increase of VCAM mRNA in the cerebellum compared to the cortex of these knockout mice. And if you, in culture, you can add interferon beta and it downregulates VCAM production. Okay, so the idea is that we're not getting enough interferon made because the receptor is compromised, and so we've got higher VCAM and higher trafficking into the CNS.
Right, so VCAM is grabbing the cops and pulling them in the door.
They are. And the astrocytes are important for regulating that.
Right.
because astrocytes make interferon normally. Interferon downregulates VCAM expression, so you get less infiltration. What else here? They blockade VCAM. They have a compound that can blockade the molecule. It's an antagonist called Bio5192. They give this daily to the mice following intracerebellar infection. They get extended survival in the knockout mice. That shows that there's a defect in the regulation of VCAM by not having IFNAR in these astrocytes. Significantly reduced. So the antagonist significantly reduces infiltrating lymphocyte and macrophages in the cerebella of these IFNAR mutant mice. They also reduce the number of cells undergoing apoptosis. So that's a part of the problem here, that in the cerebellum you're not making enough interferon, and so VCAM is not properly regulated, and cells are coming in, and that's a problem.
And by the way, if you're having a little trouble following this while listening to the episode, this paper is open access. It's in the JCI, and it is an extensive and elaborate paper, so don't feel bad. It takes everybody a while to keep track of this. Of course,
most people are listening on their exercise bikes or in their cars, and they can't listen to the paper, so we just have to do a better
while they're
job.
Doing a plaque acid.
That's right, so we have to just explain it to them. Let me summarize this. Blockade of VCAM in these mutant mice after infection decreases inflammation and immunopathology, specifically in the cerebellum, okay?
Right.
And this means that enhanced expression of the things that enhance leukocyte migration and the concomitant neuroinflammation contribute to tissue injury and mortality following infection. Then they ask whether interferon signaling in astrocytes from both places, from the cerebellum and the cortex, impacts BBB function. So they make in vitro BBB cultures. They have primary brain microvascular endothelial cells, co-cultured on inserts above astrocytes, which are derived from the cerebellum or the cerebral cortex. So the endothelial cells are on top of astrocytes from either the part of the brain. And then they measure the integrity by measuring electricity, transendothelial electrical resistance, which is a function of the brain barrier. So I guess, Kathy, when you have a good barrier, you have higher resistance, right?
Correct.
And when the barrier opens up, more electricity goes through. That's a simple way of looking at it, okay? Addition of interferon beta to the bottom chamber results in an enhancement of electrical resistance, right? It's tightening up the blood-brain barrier when you add interferon. It's a beautiful assay. I love it how they do that here. And this is in cerebellar astrocytes. And they see more of an effect compared with cultures from the cerebral cortex. So the cerebellar astrocytes have a greater impact on the BBB in these in vitro cultures. The next experiment, they want to know if astrocytes derived from the two brain areas can influence the ability of West Nile to cross the intact blood-brain barrier. So again, they use these in vitro cultures. In this case, uniform cultures of wild-type brain microvascular endothelial cells above either wild-type or IFNAR astrocytes from either part of the brain. Lots of variables here at Dickson. Sorry. Then they put virus in the top chamber. They leave it there for 6 hours, and they see if it can get to the bottom chamber. Basically, instead of using electricity, they use virus now, because you could argue electricity is not a virus, and you'd be right. So they find less viral trafficking to the bottom of cultures with wild-type cerebellar astrocytes compared to wild-type cerebral cortical astrocytes. But they didn't see this difference when the astrocytes were deficient in IFNAR. So pre-treating the cultures with interferon limited viral trafficking, and that's consistent with everything we've said so far. Interferon is going to help tighten up the junctions and downregulate VCAM and so forth. Interferon suppressed viral trafficking better in cultures with wild-type cerebellar astrocytes than in cerebral, and the difference is lost when you lack IFNAR expression. So, of course, you take away IFNAR, you can't respond to interferon beta. That makes total sense. So you got it, the IFNAR.
Got it.
Interferon is tightening up the junctions in these in vitro cells, and which would normally let West Nile virus in. Okay, so those are the main experiments. The last ones are all concerned with looking at the gene expression differences in these two regions of the brain. They take astrocytes derived from the two parts, and they do RNA-seq to look at what is made in them. And in fact, all the molecules that we've talked about so far make sense. They see when you compare the transcript levels, for example, they see enhanced basal expression of several ISGs.
Interferon-stimulated genes.
Correct.
Yes.
And in contrast, basal expression of pattern receptor recognition and other signaling molecules don't change between the two types of astrocytes. So, Kathy, did you notice this sentence isn't quite constructed properly? Say, remarkably, we observed enhanced basal expression of several ISGs, blah, 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 but they don't say enhanced in where, but I assume it's cerebellar compared to cerebral.
I circled it and asked at first in cerebellar ? And then
Right.
I found it from the figure, yes.
In the figure you can find it, yeah, but not in the
Yes.
text, right?
Yep, I had that same problem.
Got it. Yeah, I read it like 4 times, and I said, "What?" Because
Yeah.
there are a lot of
Compared to
molecules
what?
listed. There are a lot of molecules
Right.
listed in parentheses, so it gets a little hard. So basically, astrocytes from the cerebellum are primed with enhanced interferon signatures compared with those from the cerebral cortex, and they get maximal ISG expression more rapidly when you add interferon.
Now, I did have a question about that too. I circled the word remarkably and put a question mark by it. I didn't.
Not remarkable, yeah.
It's kind of like, wouldn't you expect something like that? But
Yeah.
so maybe I'm missing something. But yeah, okay.
Well, I think what they're leading up to is this discussion about how the astrocytes, same cell type but in different parts of the brain, are primed very differently for different types of activities.
Oh, maybe the remarkably is going to refer actually to the next sentence. So first of all, they observed these basal expression differences in the cerebral cells.
Mm-hmm.
But then in contrast, basal expression of some other molecules didn't differ between the two types of astrocytes.
Right.
Data not shown. Okay. But so then maybe that's where the remarkably should have been. I don't know. It just threw me. But okay.
So basically, many people have always thought that there's a uniform picture throughout the brain, but it's not the case. Here there's heterogeneity. They call it immunologic heterogeneity of the CNS. The astrocytes as we've seen in the cerebellum, are not behaving the same as in the cerebrum.
So I'm going to ask the dangerous why question.
So they actually look at that very briefly, and they say it may be conserved, a phenomenon that may be conserved in recent evolution that perhaps serves to protect centers of vital autonomic function in the cerebellum and brainstem from pathogenic and immunologic injury. So they're saying the cerebellum is more important maybe than the cerebrum.
Well, we know that to be true, right? Because clearly thinking is not really
Is not valued.
not valued, right?
You lop off a whole bunch of...
Yes.
As long as you can walk around and swing your arms and stuff like that.
So basically,
Breathe,
your breathing and your heartbeat,
That's right.
they're important to conserve, and so they're
Right.
protected
It's the
differently.
autonomic nervous system.
But here the thing is, why not protect the whole brain as well? Maybe there's a downside to protecting the cerebrum. Maybe it costs, energy costs, a lot of things. I know you can't do whys, but it's an interesting issue. I thought of the same thing. Why the cerebellum?
Maybe it can there was a whole bunch of studies done once with regards to toxoplasmosis, which also attacks various parts of the brain. And the brain responds by actually shifting functions. When it loses it in one place, it shifts it to another place. And these kids that grow up with congenital toxoplasmosis into young adults that make it that far, there was one girl that I recall seeing on a Grand Rounds presentation once that was absolutely stunning. Her brain cavities, the spaces between the brain tissue, were enormous. I mean, she had almost no brain tissue left, and yet they said for her to go to the board and to write out the quadratic equation. And half of the audience panicked because they thought they were going to be asked the same question. This girl gets up and with very thick glasses because she had chorioretinitis as a result of her illness, she perfectly wrote out the quadratic equation at the age of 12, where her brain had regained function where tissue was missing.
Right, and that's
quite
that's transferring functions probably within the cerebral cortex.
Yeah, the ventricles is what I was trying to say.
Right. Well, where on the other hand, if your cerebellum gets screwed up,
you're dead.
You can't transfer those functions to the cerebral cortex because you will have stopped breathing.
Here.
So it's a critical area to protect, and I suspect, Vincent, that you hit it on the head when you mentioned the energy cost.
No pun intended, of course.
Yes.
Yeah, right.
It takes a lot of energy to mount this kind of a hypervigilant defense, and the brain is already and the cerebral cortex is already demanding immense amounts of energy. So I suspect it's something like that the selective pressure is just not in favor of having this high energy type of defense universally. So you protect the stuff that would kill you immediately if it gets infected.
Yeah. Now, so the bottom line here is the astrocytes are different in the two regions, and their key role is early in a virus infection, there's inflammatory cytokines produced that open up the BBB, let cells in, and the astrocytes, through an interferon-mediated response, dampen that to keep it tight, and they do that through VCAM1. Now, the cool thing here, not so cool, but interesting, we could not do these experiments with polio, because if I put 10 to the 2 PFU of polio in the foot pad, nothing would happen.
Right.
The reason it works for West Nile is because there's local replication in the foot pad in a variety of other cells, and you get the high titers you need for brain invasion. But for polio, even in transgenic mice, it doesn't replicate there. It only replicates in the brain. So that's a limitation of an animal model.
So you're going to assume also that the death of these mice for the cerebellum was a bystander effect of just inflammation from invading cells?
Well, I think the
not from the virus itself?
Well, the virus is killing neurons.
Yeah, but these animals died quicker because the cells were able to get in fast.
Well, I think yes, exactly. If you take away the interferon response in the astrocytes, then the barrier's open, virus get in more readily and kills them faster. Yeah. Okay. Now, they say that people, their results with blockading VCAM suggest that people with encephalitis, so apparently there are people with encephalitis and persistent cerebellar inflammation. So it's not acute. They might benefit from VCAM blockade, which is an interesting idea. In other words, block VCAM so that you stop getting the infiltration of cells into the CNS. It would help close up the flow.
Right.
Interesting idea. I remember once, Dickson, early on in the podcast series, you said, treat patients with West Nile encephalitis with steroids.
Right.
And I told that to Michael Diamond once. He said, oh, no, gosh, no, you would never do that.
Exactly.
But the idea would be to suppress inflammation, right?
That's right.
But now I forgot why he said. In some cases, it works, but not for West Nile.
Yeah. Well, as
going back to a question that came up very early in the this paper about the pathogenesis of the virus, I looked it up on CDC site. 70 to 80% of people infected don't have any symptoms.
That's right.
There's febrile illness in maybe 20%, and less than 1% develop a serious neurological illness, and 1 in 10 of those can have permanent neurologic effects or die. So we're talking 1% of people who have neurologic effects and 0.1% who get in worse trouble.
And mostly older people.
And because there's so many infections annually in the U.S., that amounts to thousands of cases. And a lot of them have long-term sequelae.
Yes, that is true. That is true.
So that's why it's not a good thing. In fact, in general, I would say having a virus in your CNS is not a good thing.
Generally not a good thing. No.
Whether it be West Nile or measles or polio, it's not a good
rabies.
Thing. So it's like Kathy was saying, this really good block, even down to small molecules, to getting into the CNS. And that keeps a lot of viruses out. But just a couple of them can get in, right? Polio and West Nile and herpes viruses. And when they get in there, they wreak havoc.
They do.
And we'd love to know how that works. I mean, this work is part of an answer.
Yeah.
But in addition to using that to combat those viruses that get in, this would be really, really useful information for drug development. Because a major problem with something like a protein-based drug is you're never going to get it past the blood-brain barrier.
That's right.
So there's a whole, most of the flavies are able to get across the blood-brain barrier. All the eastern and western equine, lacrosse, St. Louis, West Nile. There's very few that you can name that don't do that. I guess dengue is probably one of those.
I saw recently some reports of dengue even getting in.
Really?
So I wonder what it is that they're able to do.
And that's a whole, there's got to be some commonality there, yes?
Mm-hmm. I would guess.
Sure.
I see you've put in some identification of the choroid plexus, Kathy.
Yeah, so I think in the original picture that we had, it's probably that darker aqua region to the right.
Mm-hmm.
Right.
Right. I was trying to find it all this time, and I think my Atlas of the Mouse books are out in the vestibule. So I found some things online. Not only do viruses, some of those flavies that we were just talking about, get in by maybe disrupting the blood-brain barrier, but there's also this thought about the Trojan horse kind of thing. So that's...
Getting inside of a cell, you mean, yeah.
That's right.
Yep.
Right.
Absolutely.
And of course, as I think we touched on up toward the beginning, there are viruses that bypass this whole thing by invading peripheral nerves and then transferring themselves through or getting transferred through the nerves themselves into the nervous system.
And don't forget the trypanosomes, because they get into the ventricles. They don't get into the tissues, but they cause havoc with inflammation once they get there.
So do they cross the
Good
brain
question.
CSF barrier?
Well,
is that why you were asking about the
I think that's the way
plexus?
they get in. I think they get in through the choroid plexus rather than through the capillary system.
So the dengue virus neurovirulence I'm thinking of is in mice. You can adapt it to be neurovirulence. I'm not sure that it's ever been associated with human neurovirulence. I don't want to misinform people. I don't want to make incorrect statements. I don't want to lie because
I don't either, but I continue to do so anyway.
As an author says later on in our show notes, lying is abominable.
And I just want to say, because we may not be including this figure in the show notes, the darker aqua region, kind of where we think the choroid plexus is, kind of underlies the cerebellum,
right,
toward the back of the brain.
Yep. Which in a mouse is, well, this is elongated, of course, right? It's a little different from people, but in both the cerebellum is in the back, right?
That's right.
Yep. All right.
Correct. It's protected by thick bone.
Try and get Dickson to refresh his screen, and then he'll see these additional pictures.
Dickson, there's a little refresh squiggly thing at the right hand of the address bar.
Circular arrow.
No, I know.
It's an arrow on a circle.
I know
you
where
know
you
about
should.
that.
I see it.
All right, let's do a few emails here.
Just did it.
Just a few. Maybe one round.
Thanks, Kathy.
Time is getting long in the tooth. Is that the right expression?
Something like that.
Richard writes, just got my new T-shirt. Thought the TWiV crew might be interested. Thanks for the great podcast. And he sends a link to a website called viralstyle.com.
Yes.
And his T-shirt is, "Science is not a liberal conspiracy."
And I promptly linked on this and ordered one for myself and one for my wife, and we'll wear them to the march.
All right.
Dickson, you going to the march?
You betcha.
Oh, good. Yeah, we are going to go too.
We're all going to go.
By the way, I saw a bit. I guess we circulated this amongst ourselves. I guess I forget that PLOS Pathogens has in effect endorsed the march. Said they'll be there.
Nice.
So future Senator Michael Eisen will presumably be there.
So maybe Harold Varmus can be our champion.
Harold might go.
He invented the
PLOS. He's a political guy.
I think they ought to get Neil deGrasse Tyson to speak.
Hear, hear.
I think they should get Rich Condit to speak.
What would you say if you could speak, though, Rich?
Or Bill Nye.
Bill Nye?
Just that lying is disgusting.
That's right.
Dickson, Dickson, can you read the next one?
This is Alex.
Alex, yeah.
Alex writes, "Hello, TWiVsters. I was shocked to hear that the 17th email still hasn't come in for TWiV 423, so I'm throwing my hat in the ring, fingers crossed. Also, even if get beat into by number 17 by another listener, I just wanted to say thank you for your podcasts. I'm a graduate student and now fairly a long-time listener to TWiV, TWiP, and TWiM, and have probably passed hundreds of hours of bench work with you folks in my ear. Thanks for all you do. All the best, Alex.
Cool. I don't know which contest that was. I think that was the previous one.
So, maybe 17, when we have one of these and we finally get to the number X listener writing in, then all the subsequent ones, we can just enter them in the next contest.
No, that's right. Just play it forward.
Think so too.
Play it forward.
But now it's too complicated for me because this book is already gone.
Let's write another one.
Rich, can you take the next one, please?
Sure. L'Oreal writes, "Hello, TWiVers. I just downloaded episode 425 and heard you hadn't received enough emails yet for your Infections of Leaves Your book giveaway. I imagine I'm too late at this point, but figured it was worth a try anyway. The weather in the Research Triangle has been decidedly spring-like since the last bit of snow melted. Currently 56 degrees Fahrenheit, 13 C, clear skies, with a scurry of squirrels at the new bird feeder." Somebody once this is an aside, somebody once gave us a really cute bird feeder, this in Buffalo, made of wood, with glass sides and stuff. And the squirrels not only ate all the bird food, they ate the bird feeder.
Really? Oh my gosh.
They ate the whole thing.
Good heavens.
Unbelievable.
Oh, wow.
Looking forward to Vincent's promised spring trip to our area soon. Wish I could make it to ASV this year, but keeping my hopes up for next year. P.S. I should have listened to the whole podcast before sending an email. The AAV capsid is composed of 3 structural proteins called VP1, VP2, and VP3 in approximately a 1:1 to 10 ratio. VP1 contains a PLA2 domain, and this is characteristic of all parvoviruses. In the case of AAV, the infecting virion undergoes receptor-mediated endocytosis, and the acidification of the endosome causes the internalized N-terminus of VP1, which contains the PLA2 domain, to be extruded through the 5-fold pore of the capsid. The PLA2 activity then releases the virus from the endosome, where it can finish trafficking to the nucleus. You should do an AAV show soon. Thanks for the great podcast. So, we mentioned that, but that's a much more complete and accurate description of how the parvoviruses get out of endosomes for their ultimate trip to the nucleus. And I would imagine at ASV we will hear more on TWiV about parvoviruses from Mavis.
Right. So this was about TWiV 425, where PLA2G16 was this protein that they identified, which is a switch between entry and clearance. And they noted that this PLA2-like domain also is in the AAVs, the parvos, right? So that's what that's about. Kathy, can you take the next one, please?
Yes. Michael writes, "Dear TWiV hosts, first I am enjoying both TWiV and TWiP. I'm going through the archives of both shows and wish I had all you as professors when I was a medical student, though I doubt any of you are old enough."
Dickson is.
I'm definitely old enough.
I saw a patient yesterday whose mother told me that after having the hand, foot, and mouth virus last summer, all his warts had fallen off. I was incredulous, but we had tried various measures for many months, and all it took was a good Coxsackie infection to rid him of all his warts. I had never heard of this before in all my years of practice, seeing both conditions many times. Have any of you? Thank you, and best regards, Mike. So, Vincent, I guess you asked Daniel Griffin about this. And Daniel wrote, "It makes sense. Just getting the immune system to respond is often what is required to clear the virus causing the warts. I must admit that I have not seen it or read about it, but I would believe it." And then someone's inserted a link from Lewis Thomas. Good, Rich. It's called On Warts. And it describes kind of the same thing, that you could just think for the warts to go away, and they will.
You can hypnotize people to make the warts go away. This is described by Lewis Thomas on this essay. So I think that essay was a pick a week of mine years ago. But it starts with doctors pointing out that they can do irrelevant paint on gentian violet, which does nothing. Okay, but it convinces the patients that their warts are going to go away, and they go away. Okay, and ends with this observation that you can hypnotize people. You can you can actually hypnotize people and tell them to get rid of warts on one half of their body. And they'll do that.
It. Oh, no. I'm going to draw a line here.
I'm sorry. Read the essay. He's documented this. This is Lewis Hum. And some people, as typically people do, get mixed up and get rid of the warts on the wrong side of the body.
Oh, my God. That's a tragedy.
So there you go.
But this placebo effect actually explains why there are so many traditional remedies for warts.
Yeah.
Because anything works.
Right.
And as Lewis Thomas kind of concludes at the end, something to the effect that Daniel did, something about activating the immune system may do the trick.
Yep.
Vincent, I had a question. Hand, foot, and mouth virus is some kind of picorna, but not necessarily a Coxsackie. Is that right?
Well, Coxsackie can cause hand, foot, and mouth disease, right?
Okay.
But I think other viruses can cause it. I mean, you basically have a rash on your hands, on the palms of your hands, the bottom of your feet, and in your mouth. And
yes,
the name.
My grandson had it just recently.
And with Coxsackie, it's a common one. You get a very high fever, very typical conglomeration of two symptoms. Yeah, and it's very interesting. That makes sense, though, that it would activate it, and it's cool. I didn't know this, but Daniel, I figured he might know since he's our resident physician on TWiV. All right, Alan, can you take the last one there from Anthony?
Sure. Anthony writes, starts with a link to a posting on Facebook from the American Fancy Rat and Mouse Association. January 28th.
Without that, we wouldn't have mouse genetics.
That's right.
Yeah, good point.
Laugh all you want, but...
So January.
I'm laughing at the fancy part. That's all.
The fancy.
Well, it's called the mouse fancy and the rat fancy, and there's a cat fancy, and that's.
I know they're also
weird.
There are also pigeon breeders too that have done a lot for pigeon genetics.
And in fact, if it weren't for pigeon fanciers, we wouldn't have a good section of Darwin's Origin of Species.
That's right.
This is all true.
That's right.
That's a very, very significant set of data that he got from pigeon fanciers. Anyway, the American Fancy Rat and Mouse Association, which is a very serious organization that is not funny in any way,
You bet.
says on January 28th, 2017, show canceled. In light of the hantavirus outbreak in the Midwest and with the CDC working their way west to find how far the infection has spread, we're canceling the January show to keep everyone's rats and their owners safe until we know the final results of their testing. Anthony adds that the post at Facebook contains links to the CDC, Illinois Public Health, and a news story website. And says, by the way, if memory serves me correctly, in one of the chapters in Morse's Emerging Viruses, there was speculation that a diverse range of ailments often found in impoverished U.S. neighborhoods were due to some unknown hantavirus. I've been wondering if anyone went on to look for this.
Yeah, this is an outbreak of hantavirus, which is, if everyone may be familiar with the sin nombre hantavirus, which is...
But what is it called?
Yeah, right.
Without a name.
And it's host to the deer mice, and people get it from deer mice, and they get a very serious respiratory disease. Now, the mice are okay. In this case, this is a hantavirus that causes high fever and kidney problems, different clinical syndrome, and the reservoir is rats. And again, rats are fine. So apparently there's been an outbreak in the Midwest. People have been buying pet rats, and they're getting infected. And, this term rattery has been coming up now because that's where you breed these rats in ratteries, right?
Oh, yeah.
And the pet dealers they've been going in and they find seropositive animals, and that's how it's spreading. So apparently...
Well, infection is the sincerest form of rattery.
Oh!
It's a title. It's a title. All right, we'll stop there because the next one is quite long, and we know you're almost done with your plaque assay. So let's do
That's right.
couple of picks.
I don't think I could do a plaque
work.
assay. I don't think I could do a plaque assay and listen to TWiV at the same time. I
can't work and listen to things. The only thing I can do is drive. I do listen to podcasts while driving, but it's funny. I cannot listen to an audiobook when I'm driving.
That's what you've said. Yeah,
I can't concentrate enough. I tried listening to things.
I think I could overlay an already established plaque assay listening to a podcast, but that would be about it.
We listen to old broadcasts of Bob and Ray and get hysterical and almost drive off the road, but we find that highly entertaining when we're taking long trips.
Dickson, do you have a pick?
I do. I have a pick that's very interesting, actually. It's a house made out of shipping containers. It's called Honomobo. And it's a site you'll love this when you click it, because a shipping container is about the most homely of all containers. They fill them up, they bring them across the ocean, and then they leave them in the port cities because they're too heavy to take back. So Newark, for instance, has hundreds of thousands of shipping containers just piled on top of each other, right? Huge. So people are racking their brains as to what to do with them, like old tires that don't have any tread on them any longer. So a lot of them have been turned into these grow systems that we've been talking about.
That's right. Yeah.
But this website really attracted my attention. And when you click on it, you will see some of the most modern, beautiful, almost Finnish architectural style homes that everybody would want to live in one of these.
Yeah.
And it must be cheaper than dirt to start to assemble, but the internal components, of course, cost a lot of money.
Yes.
Yeah, the granite countertops and the fancy sinks.
The point is that getting this stuff up and running would cut the time of construction in half, at least, maybe even more than that. And they tend to stack up nicely, and you can put them in a lot of different places. And
I don't know. Most people aspire not to live in a box.
Yeah, really. I mean, I think in the end, you've got so many windows and so much other stuff. I'm not sure it's much cheaper than prefab walls.
I don't know if I didn't see any prices on them, so I was just attracted to them.
I can almost guarantee, having worked summer jobs in construction and having had some home renovations done,
I've done some
that this is not a bit cheaper than
but they're beautiful.
Traditional construction method, because the money in a house, the money for the wood in your house is nothing. It's really, I mean, it's just.
I just meant time. I didn't mean time of expense. I meant
even that. Framing up a house happens almost instantly. What runs up the money is the plumbing fixtures and the electricity and the heating, ventilation, and air conditioning system, and that's where the.
But maybe it keeps the containers from going to the landfills?
If it's a better use for the containers, then that's a good deal. On the other hand, these containers are generally made out of steel, which seems like you could scrap that.
Right.
They don't.
That's right. I don't know. I drive past a pile of them every day.
Oh, yeah.
On the New Jersey Turnpike. Huge. I mean, it must be 20 stories tall and a mile long. They have a big sign on the side for sale. Come by, please.
Take my container, please.
They should pay you to take them away.
Right. No, I just thought it was a wonderful iteration.
End up getting bought and shipped as their original purpose.
Yeah,
they have a link on this site called Why Shipping Containers, and they go one of the lines in this is that with more boxes being imported than exported, we have an opportunity to reuse something that is desperately looking for a new home. And they go into the fact that they're constructed to be both modular and extraordinarily durable.
Right. A lot of them are off the grid because they're, they got solar panels and that sort of stuff. So it's.
But these things basically they can go right onto a truck, right, or on a railroad car.
They're certainly easy to transport. They're built for that.
Yeah.
Right.
Well, they should make them of material that self-destructs. So wouldn't have to worry about it.
Well, that's what we do with our houses already.
Alan, what do you have?
Well, Valentine's Day is coming up, so I have a site that's ideal for shopping for a gift for that special someone. Well, depending on who your special someone is and what their tastes are. But anyway, I find this site very useful. This is Skulls Unlimited.
Oh my gosh,
love it!
Have you dropped the hint to your wife, Alan?
Well, no, I've actually. I've placed an order here that hopefully is arriving today, so I'll have time to gift wrap it.
You better not hurry because you have to be long in the tooth in order to make this happen.
Right. Yes.
Wait, is this something that you want or that she would want?
That she would want.
Okay.
I certainly appreciate the stuff too, but she definitely likes. I don't know if you've seen the background in my office on any of the videos. There's some stuff on the the mantelpiece behind me. That's looks like you might find it in a Victorian Natural History Museum, and it comes a fair amount of it has come from this place. So this is just a nifty place to shop around, even if you don't end up buying a skull or a fully articulated skeleton of whatever species you're looking for.
They have both real and fake artificial skulls, right? They have a T-Rex skull.
Right. They have the fossil stuff is all casts.
Yeah,
but they're museum quality casts that you might see on display or that you might use in an anthropology class.
Dickson, the T-Rex is only $9.
Really?
How big can this be?
Not big.
They have miniature ones as well.
So there's a place in New York City called Maxilla and Mandala, Mandible and Maxilla.
Very, very similar.
And
right.
Got lots of interesting things.
This is pretty cool.
It is.
You can get a human skull for a mere $1,800.
Look at that.
Yes.
That's a real one, right?
That's a real one.
Some years ago, I was really quite tempted to get myself a human skull. Oof. I don't know why. But the closer I got to it, the creepier it was, and I
would agree with you.
Yeah,
that used to be somebody.
That is definitely not something that I want around my house.
No,
I draw the line there.
If you needed to do Hamlet, you need to get one anyway.
Exactly. Well, right. I think I would just get a cast for that.
Yeah,
because you need to get a cast for a play anyway.
That's right. Well, you could always have them around. Have them around for skull sessions.
This is very cool. Skulls Unlimited.
Skulls Unlimited.
That's right.
And it was started by a guy who was just into skulls. And he started a collection, and people said, "Hey, you could go into business. People would buy these." And he did, and they did. And they've got apparently now a museum of osteology that they've set up, and branch offices and all that.
Yep.
Kathy, what do you have?
I picked a link to a blog post. It's called "Why Vaccines Don't Cause Autism." And it's a good blog post written by someone who has an adult autistic child, but is, I believe, is a physician, or I'm not sure, or a PhD, and he's very pro-vaccines.
He's a physician, yeah.
Yeah, and he includes links to various papers proving various things showing various aspects of there is no link between autism and vaccines. And so I thought it was a nice compilation all in one place.
Yeah, it's very good.
And this is Peter Hotez,
there's only one real objection that I have, Kathy, and that is that you've got to be able to read in order to know what's in the paper.
Well, you could just read his blog post, which just kind of tells you why. Here are these papers that refute the link between maternal immunization and autism, for example.
Right.
So.
It's Peter Hotez, Dickson. Peter Hotez wrote.
Peter he's president of the Sabin Vaccine Institute.
I know Peter very
Yeah, that's right. I forgot to mention that.
Is he on your book, Dickson, 6th edition? Yeah.
Yes, he is.
We had Peter on TWiP, remember?
We did.
Long time ago.
And we can have him again if we'd like.
Really?
We could.
Okay.
So he knows vaccines, and he has an autistic kid, and he's laying out the evidence here just in case you wanted to see it.
Yeah.
Yeah, that's good. Sol sent me this earlier this week.
Yeah, that's very, very nice.
Rich Condit, what do you have?
I have picked one of a series of articles from Journal of Biological Chemistry, which are reflections. All of these are good. And I think we may have picked one or two of these before. These are where they have senior scientists who've had notable careers reflect on their careers. And this one was pointed out to me by my buddy Niseen Musashi, and it's by Bernie Moss, an eminent pox virologist reflecting on his career. It's entitled Investigating Viruses During the Transformation of Molecular Biology, and talks about his career. He's actually an MD, and I must be 10 or 15 years older than me, so his career goes back into what even for me was the Dark Ages, and talks about, doing... he's been there for the whole thing, the whole molecular biology revolution, and talks about his getting into pox viruses and the stuff he did with them. And it's really very nicely written and a really nice reflection. One of the things I noticed was he started working on vaccinia virus as a fellow at the NIH. He was what we call a yellow beret. He did NIH service at the NIH as alternative to becoming a physician for the Army, having received his draft notice. He figured actually that given his training, he probably would have been at a base in the U.S. working as a pediatrician. But he preferred to continue doing basic research. So he went to the NIH where you could do your alternate service, started working on viruses, and he started working on vaccinia virus, the vaccine for smallpox, in 1966 with Norman Salzman. And that was about the year that the WHO, I think it was the year that the WHO launched the eradication campaign. So the interesting thing to me is that he started working on vaccinia when it was still a very prominent public health threat. Right?
Mm-hmm.
Right. Not necessarily in this country, but globally.
Yeah. Right.
When I finally set up my own independent laboratory in 1978, 12 years later, it was the year they declared smallpox eradicated. I thought, okay, what am I going to do now? But at any rate, this is a very nice reflection by Bernie. I recommend
nice.
it.
Yeah,
and papers like this are great, and I'm glad to see that the JBC has put it online for free. These are a trove for historians.
Yeah, for sure.
Because this is a perspective that's not in any of the published papers. It's not anywhere else that you're going to find. And straight from the horse's mouth, this is somebody who didn't just live the history; he caused it. And he's telling his story. It's great.
Got to be out there. Yeah. Yep.
In his story, he references several other individuals who have written reflections. And,
yes.
Like one from Jim Darnell. That would be a very interesting read.
Right.
Okay, et cetera. So my guess is you could access the whole series and have a good time
I think so.
Of course, we had a reflection last week from Andy Ball.
Yeah.
And also we have oral reflections here on TWiV. We have people telling their stories, which is good for people who are driving or working and doing plaque assays. But all this stuff should be recorded for sure, so it's there. You don't want it to go away with the person. My pick is a very nice article, Science-Based Medicine. I've picked articles from that site many times, really thoughtful commentaries on life sciences. And this one is called Donald Trump versus the FDA. Is the standard of evidence for drug approval actually too low rather than too high? So this, among the other silly things that Trump is doing, he wants to appoint a commissioner that's going to make the FDA do fewer things. He thinks they do too much. And in this article are linked a Nature commentary called Consider the Drug Efficacy Before First-in-Human Trials, as well as a New York Times article published yesterday. And he's got 3 likely picks for FDA commissioner. All favor loosening drug approval standards and could very well undo decades of safeguards. And right now, we have lots of preclinical data required to show not only safety of a drug, but efficacy in animal models until you get into a phase 1 in humans. And the argument now is that they want to get rid of these efficacy trials in animals and just show safety and then go right into people. So we have 3 people. Of these 3, 2, one of them believes the FDA shouldn't have to require evidence of efficacy, only safety before approving drugs in devices. Apparently, the free market will sort it all out. The other believes a Yelp for drugs would do a better job than the FDA in assuring drug safety. Apparently, he's never spent much time reading online rating systems.
Right.
Yeah.
So I think everyone needs to read this to be aware of this. You may not have known that these changes are planned for the FDA by the Trump administration. I think personally that Big Pharma will fight these very strongly because they don't want to put drugs out there that are going to cause problems in people or not be effective.
This would be Big Pharma. Those folks are way more than smart enough to realize that this would be a terrible, terrible thing for them.
Right.
Not immediately, because immediately, you get, hey, rubber stamp, get everything approved, lovely. But the long-term picture is if you're a pharma company and you get a drug approved and it turns out not to work, in the current system, at least you can lay some of the blame on the FDA. Mm-hmm. If the FDA is just shoving everything through, then you're going to be on the hook, and this is not going to end well.
Right.
Can you imagine releasing a vaccine with no efficacy data and it doesn't work? It's going to give more fuel to anti-vaxxers, right?
Yeah.
Hey,
Just to name
wait
one
a minute. Wait a minute. I think you're onto something here.
What am I onto? Anyway, check it out. It's very good. I had on TWiV this week Michael Merchlinsky, who is currently at BARDA. He's worked a lot on the new smallpox vaccines and stockpiling them and antiviral drugs. And I mentioned this to him, and he said he used to work at the FDA for many years. And he said almost everyone has tried to take apart the FDA over the years. And he says in the end, it doesn't work, and he thinks that none of this is going to happen. But it's still scary, nevertheless. And as it's just crazy. He knows nothing about science, as we know. He lies left and right. And now he wants to mess with something that I think is important for us to keep things safe. So get more frustrated and infuriated. That's a thing. That's a daily thing nowadays, right?
Yes.
And Kathy has reminded us that there's a picture from the Times article of Trump with Dr. Oz. Great pair.
Yeah.
Great pair. I guess that was during the campaign. All right, that'll do it for TWiV 428. You can find it at iTunes at microbe.tv/TWiV. And also, if you want to listen on your phone or tablet, you just get an app for free and download it for free and listen away. Send us your questions and comments, TWiV at microbe.tv, and consider supporting our work so that we can do more and travel and so forth. You go to microbe.tv/contribute for all the different options. Check it out. Dickson Despommier is at thelivingriver.org and parasiteswithoutborders.com. And thank you, Dickson, for your participation, for your interest, and for your erudition.
What can I say? But I had a great time. I thought of a great title for this picture, by the way.
The one with Oz and Trump? What?
The pictorial representation of a double-blind study.
It's very good, Dickson.
It's good.
Double-blind study, yes. Oh, dear. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit, formerly a professor at the University of Florida in Gainesville, currently professor emeritus in Austin, Texas. Thank you, Rich Condit.
Sure enough, always a good time.
Alan Dove is at alandove.com, and you can find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the sponsor of this episode, Blue Apron. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.